Good day. In this lab, we will discuss one of the most important family of the gram-negative bacteria, Enterobacteriaceae. These bacteria are commonly found as a part of the normal flora in the intestinal tract. However, some species can become opportunistic and cause infection. This family includes many medically important species such as E. coli, Salmonella, Shigella, Klebsiella, and Proteus. By the end of this lab, we will understand how to identify this bacteria using culture characteristic and biochemical tests. We can identify this family of the bacteria in lab by colony morphology and biochemical tests. The biochemical tests include carbohydrate fermentation, motility test, MVIC test, urease production, hydrogen sulfide production, trouble sugar iron, and we first observe how the bacteria grow in a culture media, then confirm the identify using biochemical reaction. Most common culture media used for Enterobacteriaceae are aeugine methylene blue, Salmonella, Shigella agar, Macongi agar. All these media selective and differential. Selective, it means inhibit certain bacteria, allowing other to grow on it, and differential, it means differentiate between the species of the bacteria according to the biochemical characteristic and usually lactose fermentation. Now, the aeugine methylene blue agar is selective and differential for Enterobacteriaceae gram-negative bacteria. Is selective for gram-negative bacteria because it contains the dyes aeugine and methylene blue, and this dye inhibit growth of the gram-positive bacteria, allowing growth of the gram-negative bacteria. Why it is differential? Because it contains the lactose, which can differentiate between the lactose fermenter from the non-lactose fermenter bacteria. And this media is important for identifying the Escherichia coli. In this image, you can see the growth of the E. coli on this type of the agar that lead to produce dark blue-black colony with a metallic green shine, and this metallic shine is important for identification of the E. coli. Why this character is mostly specific for the E. coli? As I mentioned, it's a selective for gram-negative bacteria. It allows a gram-negative bacteria to grow on it. And it contains the dyes aeugine and methylene blue. E. coli, when grow on this type of the media, it lead to the fermentation of the lactose and to produce large amount of the acidity. This large amount of the acidity, it react with this dye and lead to the color change, which produce the dark blue-black column with a metallic green shine. Other gram-negative bacteria, such as those that are non-lactose fermenter, will lead to the colorless when grown in this type of the media, while the Klebsiella that are also ferment the lactose, why don't produce the metallic green shine? Because it ferment the lactose while produce small amount of the acidity, and this small amount of the acidity when react with this dye lead to the pink color, not black blue colony with a metallic green shine, which is a specific characteristic of the E. coli. So both of them, E. coli and Klebsiella, is a lactose fermenter, but the E. coli is a strong lactose fermenter and lead to the production large quantity of the acidity. This acidity react with a dye and lead to the blue-black colony with a metallic green shine. Another selective and differential media, Salmonella, Shigella agar, which is mainly used for stool culture. For identification, Salmonella and Shigella is selective as it inhibits growth of the gram-positive, allowing the growth of the gram-negative. Differential, it contains the lactose. It can differentiate the lactose fermenter from the non-lactose fermenter bacteria. In this type of media, we can also detect the production of the hydrogen sulfide, which can detect as a black center. Salmonella and Shigella, when grown on this type of media, produce the colorless colony because it's not fermenter bacteria. But Salmonella produce colorless colony with a black center, while Shigella without a black center. Why? Because Salmonella release the hydrogen sulfide, while Shigella without hydrogen sulfide. The coliform colony, like the E. coli colony, appear as a pink or red color because it's lactose fermenter bacteria. But this type of the bacteria, they are non-release hydrogen sulfide. So the colony of this bacteria without the black center. And in this image, you can show the difference between the colony of the gram-negative Enterobacteriaceae on this type of the media, Salmonella Shigella agar. E. coli can grow on this type of the media because it's selected for gram-negative, produce the red to pink colony because it's lactose fermenter bacteria without black center. Salmonella produce the colorless colony because it's not lactose fermenter but with black center due to the release of the hydrogen sulfide. Shigella produce colorless colony without black center because they are non-lactose fermenter and not release the hydrogen sulfide. Another important selective and differential media for gram-negative Enterobacteriaceae, McCongie agar. This type of the media is selective because it contains bile salt and crystal violet that inhibit the growth of the gram-positive and allowing only the growth of the gram-negative like Enterobacteriaceae. Why it's differential? Because it contains the lactose and neutral red indicator. This type of the media, we can differentiate between the lactose fermenter bacteria from the non-lactose fermenter bacteria. As the lactose fermenter bacteria like E. coli, Klebsiella, and Enterobacter, when grown on this type of the media, it leads to the fermentation of the lactose and change the color of the indicator neutral red and produce the pink colony. While the non-lactose fermenter bacteria like Salmonella, Shigella, Proteus produce the colorless colony because it not ferment the lactose in the media and not change the color of the indicator. And in this image, you can see the lactose fermenter bacteria on McCongie agar, E. coli, and Klebsiella. Both of them produce pink colony, as both of them is a lactose fermenter bacteria. It ferments the lactose in the media and lead to the production of the acid, and this acidity is lead to the change the color of the indicator neutral red and produce the pink colony. But here in this image, you can see the difference between the colony of the E. coli and the Klebsiella and McCongie agar. Klebsiella usually have a mucoid colony due to the lactose fermenter and capsule production, while the E. coli on McCongie agar appear as a pink dark colony, usually flat and non-mucoid, while the Klebsiella have a mucoid colony due to the capsule. Now, after finishing the culture media, which is selective and differential media for Enterobacteriaceae, we go to the biochemical test. The most important biochemical test, INVIC test, is abbreviation representing 4 biochemical tests used to differentiate Enterobacteriaceae: indole, Mathia-Red, Fulcus-Proskauer, and Stray. And these tests are specially useful to differentiate E. coli from Klebsiella and other coliform. Principle of the indole test. It indicates 4 tests. It indicates the indole test used to detect the ability of the microorganism for production of the indole from the tryptophan. And Mathia-Red detect the ability for microorganism for acid production from glucose fermentation. B. Fulcus-Proskauer, it detects the ability of the microorganism to production of the acetone from the glucose. And C. It detects the ability of the microorganism to use acetate as a only carbon source. About the second I in the INVIC test, it's extra just to make the name easier to read and don't represent any test. Now I will go to explain each test. In the indole test, we use to detect the ability of the microorganism for production the indole from the tryptophan, which is amino acid. We used the COVAX reagent and the media Pepton water. First, we inoculated the bacteria to be tested into the Pepton water broth and incubate at 37 Celsius for 24 hours. After incubation time, add 0.5 ml of the COVAX reagent and shake gently. And the result observed as a positive or negative. The positive result appear as a red ring in the upper layer of the broth, while the negative is yellow ring. Why this happen? The bacteria that have ability to break down the tryptophan in a media to the indole by tryptophanase, it produce a pink ring like E. coli, while bacteria like Klebsiella, they don't have the tryptophanase and don't break the tryptophan in the media and appear as a yellow ring. Methyl red test. In this test, we use the methyl red reagent and media glucose phosphate peptone water. Why we use glucose phosphate peptone water? As I mentioned in this test, we use to detect the ability of the microorganism to completely ferment the glucose in the media and produce large quantity of the acidity that maintain the pH of the media below 4.5, like Escherichia coli, that have the ability to ferment the glucose, completely fermentation of the glucose and produce large quantity of the acidity that lead to the red color. Other, like Klebsiella, it not ferment the glucose completely and not produce large quantity of the acidity. It ferment the lactose but not completely and not produce large quantity of the acidity, and when we do this test it produced the yellow color. The procedure of this test, we inoculated the bacteria to be tested in the broth media. After incubation period at 37 for 24 hours, we add 5 drops of the methyl red. The positive result, like E. coli, appear as a red color, which indicate complete fermentation of the glucose and production large quantity of the acidity, while the yellow color, it indicates the negative result like Klebsiella. The third test in the INVIC test, Voges-Perls scour test. We use this test to detect the ability of certain bacteria to ferment glucose incompletely and produce the acetone. After add the reagent A, 30% of 40% of the KOH, and reagent B, 5% of the alpha naphthalene. Acetone is oxidized to diacetyl and then react to produce the red color. The media used in this test is like that used in the methyl red test. It's a glucose phosphate peptone water. First, we inoculated the test organism into the broth media. After incubation at 37 for 24 to 48 hours, we transfer 2.5 ml of the culture to the clean tube. After, we add 6 drops of the reagent A, KOH, and 2 drops of the reagent B, alpha naphthalene. We mix gently and wait for 10 to 15 minutes. The positive result is indicated by a pink to red color, which is mainly indicated Klebsiella pneumoniae, and the negative result, they are no color change. Example of this bacteria is Escherichia coli. We use this test to detect the ability of certain bacteria to ferment the glucose incompletely and produce the acetone. E. coli usually is negative for this test, while Klebsiella is positive for this test and indicated the positive result by a dark red color, while the negative by brown color or no color change. And in a fourth test of the INVIC test, citrate utilization test, is used to detect the ability of the bacteria to use citrate as a source of the carbon and energy. Not all bacteria have ability to use acetate as a source of the carbon and energy. We use this test to detect which bacteria have this ability. We use the media Siemens citrate agar slant that have the bromo-3-mol blue indicator. And if the organism use acetate as a source of the carbon and energy, the acid will be product and cause the media to change from green to blue. The procedure of this test, we inoculated the organism on the citrate agar slant by stabbing and streaking method. After incubation, we observe the result. After 24 hours at 37 Celsius, we observe the result. The positive result is indicated by a blue color with growth. And the most common bacteria that have the ability to use a citrate as a source of the carbon and energy is Klebsiella pneumoniae, while the negative result indicate by green color with no growth, like E. coli, because they don't have the ability to use acetate as a carbon source. And that's all about this test. Here there are some video links. Watch this video to understand how these tests perform in a lab, step by step. At the end, we concluded that we use the impact test to differentiate between two important bacteria, E. coli and Klebsiella. Escherichia coli is positive for indole and methylred, and negative for Voges proskauer and citrate. Klebsiella is negative for indole and methylred, and positive for Voges proskauer and citrate. And that's all about this.